Okay, good. So today we are going to look at blood physiology. Last week we discussed the body compartments and body fluids, and we talked about the intracellular compartments and the extracellular compartments. So we talked about the extracellular fluid, and we mentioned plasma and interstitial fluid. So today we're going to be looking at that plasma. We're going to be talking about blood as we discuss the extracellular compartments. So now I'm going to share my screen. I'm going to share my screen with you, and we would go into the lecture. Share screen. Good. So you can see my screen, I believe. Blood physiology. Blood physiology. As we know, every human being has blood on their inside. Every human being has blood on their inside, and without blood, there is no life. Life is present. Blood is actually the life that we have, because without blood, there is no life at all. Why? Because every nutrient, everything, every hormone that you have in your body is transported through the blood. So blood is a special type of fluid, connective tissue. Blood is a special type of fluid connective tissue derived from mesoderm. Maybe you'll do embryology later. So mesoderm is an embryonic tissue from which all other, many other cells, organs and tissues develop. So blood comes from the mesoderm. Now the branch of science concerned with the study of blood-forming tissues and the disorders associated with them is called hematology. When you get to maybe fifth course or fourth course in medical school, you will do hematology. So hematology is simply the study of blood and the tissues and the disorders that are related to blood. So hem means blood and logos means study. Now, what are the properties of blood? In terms of color, the blood is bright red in the arteries and it is dark red in veins. Bright red in the arteries because it is fresh oxygenated blood containing hemoglobin. So that hemoglobin gives the blood that red color. So blood contains especially the red blood cell. You will see that it contains a pigment called hemoglobin, which gives it that red color. So oxygenated blood in the arteries is bright red, while the used blood, deoxygenated blood, is dark red in the veins. In some diagrams you would find, or in some textbooks you will find, that the deoxygenated blood in the veins is blue in color. Okay, they use blue to represent it. But in the real sense, it is not actually blue, it's dark red. When you get injured, for example, and blood is flowing out of your veins, you will not see a blue blood. So those diagrams do not necessarily give the true color of the blood in the veins. Now, blood makes up 8% of the body mass. Blood makes up 8% of the body mass. And we have said that the body mass is around 70 kg. So blood makes up 8% of that mass. Now, the blood also has a pH, potent hydrogen, the amount of acidity or alkalinity of the blood. The normal value of blood is slightly alkaline in nature, and the pH is 7.35 to 7.45. Now, this pH is very important because anything that happens that increases the pH, excessively would lead to what we'll call acidosis. and whatever increases the pH will lead to alkalosis. Now pay attention. An increase in pH leads to alkalosis. That is alkaline. The blood becomes excessively alkaline. Anything that decreases the pH leads to acidosis. So remember that a decrease in pH is acidic, an increase in pH is alkaline. So if the pH of blood, for example, drops to 4, that will be acidosis. If the pH of blood drops, increases to 10, for example, that will be alkalosis, and both of them are dangerous. So we need to maintain the amount of hydrogen ions in the blood and keep it at 7.35 to 7.45. So here we need homeostasis. And how do we maintain homeostasis? We maintain homeostasis of the blood pH through the kidney, because the kidney excretes excess hydrogen ions. So pay attention. Anytime we have acidosis, it means that the pH of the blood is very low. Yeah, below 7.35 is low. And that means that we have excess hydrogen ions, because acidity talks about hydrogen ions. And the only way to get rid of that acidity, or the major way to get rid of that acidity, is through the kidney. So what happens is that during tubular secretion, now there are 3 steps or 3 stages in the formation of urine. The first one is ultrafiltration, the second one is selective reabsorption, and the third one is tubular secretion. So in the tubular secretion, what will happen is that hydrogen ions will be secreted from the blood into the nephron, into the tubule, the distal convoluted tubule of the nephron, and then the kidney will excrete the hydrogen ions through with urine. Okay.
Yes, sir.
And when that happens, the body will conserve the amount of carbonate ions, or the amount of hydroxide ions. Basic alkaline ions will be preserved, while acidic ions will be excreted. That's how you maintain the pH. Then the reverse will happen if there is alkalosis. If there is alkalosis, it means that the blood pH is too high. Okay, and once that happens, sorry, 1 minute. And once that happens, we need to preserve hydrogen ion and excrete carbonate ions or hydroxide ions. And that still happens in the kidney. Okay, so we need to maintain this pH to have a steady internal environment. And then the blood is a little bit salty in taste. Of course, you know, it contains sodium and chloride ions, so it tastes a little bit salty. Then the temperature of the blood is around 38 degrees Celsius, a little higher than the normal body temperature, which we said should be 36.6 degrees Celsius to 37.1 degrees Celsius. But for the blood, the temperature is a bit higher than the temperature that your thermometer will give you when you measure your temperature in the oral cavity or in the axilla. And that was why, because the blood has this temperature, that was why when we were talking about temperature regulation, we said blood flows to the surface of the skin, takes heat to the surface of the skin, and that heat can be conducted away from the body, you know. And then if you want to preserve your body temperature, you constrict the vessels, the blood vessels close to the skin, so that blood doesn't flow there. And then the blood remains in the systemic circulation and preserve the temperature. Now, the viscosity of blood is 3 to 4 times more viscous than water. So blood is thicker than water, 3 to 4 times thicker than water. And the total circulating volume of blood in the human body is about 5.6 liters. Again, you need this amount, this volume of blood, 5.6 liters of blood circulating in your body for you to be healthy, to have normal function. If you get an injury and you are bleeding and you are losing blood, or somebody gets an accident and there is severe blood loss, we would have what is called hypovolemia, because the volume of the blood will decrease. So hypovolemia, decreased volume of blood, which could lead to shock, because blood would no longer, there won't be enough blood to supply your body organs because you've lost a lot of blood. So you need to, you will have what is called hypovolemic shock. And of course, you could also have anemia. So there will be decreased amount of red blood cells in the blood because you have lost a lot of blood. You will have anemia, and it is dangerous also. Good. So these are the properties of blood. Now let's go in details. The composition of blood. So blood in itself is made up of two main parts. Now, the two main parts of blood is the liquid part of blood and what we call the solid part of blood. Now, the liquid part of blood is called plasma, okay? While the solid part of the blood is called the blood cells. So in composition, blood has two main parts, plasma and blood cells. Now, plasma makes up about 55% of blood, while the blood cells makes up about 45% of blood. So when you have blood, 55% of it is plasma, 45% of it is the blood cells. And here you see blood in this test tube. Now, when blood, it was put in a centrifuge. There was an experiment that was done many years ago. Blood was put in a centrifuge and the blood was spinned around in a centrifuge machine. After that centrifugation took place, the blood in the test tube was allowed to settle, and it was observed that the blood separated into two parts, an upper part, which was the plasma, and a lower part, which was the blood cells. So the blood cells settled downward while the plasma was on top. After that experiment was done, I think it was William Harvey, if I remember very well. I think there's a guy called William Harvey who did that experiment. So he put a sample of blood in a centrifuge machine and spinned it around, and he noticed that the blood separated into two layers, an upper layer which he called plasma, and the lower layer which is called the blood cells. And this plasma, he observed that the plasma was more than the blood cells. So the plasma took about 55% of that blood specimen, while the blood cells took about 45% of that blood specimen. Now, the blood cells are of three types. So we have the red blood cells, which we'll call erythrocytes. Am I recording this?
Yes, sir. Yes.
I'm recording, right? Okay, good.
Yes, sir.
So we have the red blood cells, which we call the erythrocytes, and then we have the white blood cells, which is called leukocytes, and then we have the platelets, which are also called thrombocytes. So plasma is the liquid part of blood, then erythrocytes, leukocytes, and thrombocytes make up the solid part of blood, and each of them has, its function. So let's proceed and see what each of them do. So here with what I just said earlier, that blood has two components, the plasma and the cellular element. So the cellular element makes up 45% of blood, while the plasma makes up 55% of blood. And cellular elements are the red blood cells or erythrocytes, the white blood cells or the leukocytes, and then the platelets or the thrombocytes. So what is plasma? Plasma is a pale yellow colored liquid. It is a component of blood that holds the cellular elements of blood in suspension. So the plasma is just like you have soup, you cooked soup, right? And then you have meat in the soup. So those meats are suspended inside the liquid part of the soup. So that liquid part of the soup is the plasma, right? Like those meats are the cellular elements. So that is what happens in blood. So plasma is the pale yellow colored liquid component of the blood that holds the cellular elements of blood in suspension. Now, constituents of plasma. So pay attention to these. All of those ions that we talked about before, when we were talking about extracellular fluid, we talked about sodium ion, chloride ions, and all of those ions. They are found in plasma, although in different concentration, as we said before, when we were looking at extracellular fluid. So plasma is made up of 91% water. So it means that 91.5% of plasma is water. So the major thing that makes up the plasma is water, because we said that the plasma is the liquid part of blood, and it is liquid because of the content or the amount of water in it. So plasma makes up 91.5% of water, and then proteins make up about 7% of plasma, and these proteins are albumins, globulins, and fibrinogen. Don't forget that each of them has its function, again, which we will discuss later. So water, 91.5%, proteins, 7%, and these proteins are three main types of proteins: albumins, globulins, and fibrinogens. And then again, the other 1.5% are other solutes. Now, apart from proteins, you also have other solutes. Now, what are these other solutes that are found in the plasma? These other solutes are electrolytic ions. So we have sodium ions, potassium ions, calcium ions, magnesium ions, chloride ions, hydrogen carbonate ions, and sulfate ions. They are found in the plasma. And then we have gases like oxygen, carbon dioxide, and nitrogen. And then we also have nutrients like glucose, amino acids, fatty acids. And then we have waste products also like urea, uric acid, ammonia, and creatinine. So you see that almost all nutrients and solutes in your body are found in the blood, in the plasma of the blood. Although they only take about 1.5% of the blood, because they are very little, ion. But proteins are larger, and proteins take about 7% of the plasma, and then water takes about 91.5% of plasma. So everything is transported in plasma, including waste products. When your liver deaminates proteins, you will get urea, uric acid, and ammonia. And these waste products need to be carried from the liver through the blood to the kidney, where they will be excreted, all of these waste products. So your blood does an important job in transporting waste products to where they will be excreted. And then here we have glucose, amino acid, and fatty acids. We said that digestion begins in the mouth and ends in the small intestine. So at the end of digestion, you get glucose, amino acid, and fatty acids. Glucose from carbohydrates, amino acid from proteins, and then fatty acid from lipids. Now, at the end of digestion, these nutrients need to be carried from the small intestine into the liver for further metabolism. And that is where your hepatic portal vein comes in. So your hepatic portal vein carries these nutrients from your gut or from your intestine to your liver, where there will be further metabolism of these nutrients. And then gases, the gases you inhale through your nostrils, they get to every cell of your body through the blood. It's the blood that carries them. And then these ions that you get when you maybe eat anything containing these ions, they need to be carried also to the different tissues where they are needed to function. Okay, good. So your blood does a whole lot of work. Good. So now this diagram is showing us the constituents of blood and the function of each of them. So why is there water in the blood? What does the water do? So the water helps with absorption, transportation, and then releases heat also. So that's a function of water in blood. Albumins, what do albumins do? They help with osmotic balance. So albumin makes sure that the osmotic concentration of your blood or of your extracellular fluid is maintained at a normal level. Whenever you have a decreased amount of albumins, or you have you don't take enough proteins and you have decreased albumin, something will happen. You will have, there will be a decrease in what we call oncotic pressure, which we'll discuss this in the future. So albumins, maintain the oncotic pressure of blood. So once albumin is lacking, there will be a decrease in oncotic pressure, and a decrease in oncotic pressure would lead to a kind of edema, which we'll talk about later. And then globulins, they act to defend. They are used for defense mechanism, and there are different type of globulins, which we would also discuss in the future. And then we have fibrinogen, which helps blood clotting. In the last class, I showed you the positive feedback mechanism of blood clotting, and we talked about how prothrombin is converted to thrombin, and how thrombin converts fibrinogen to fibrin, and then the fibrin forms the clot. So this fibrinogen helps in blood clotting. And then we have the electrolytic ions that we saw in the plasma. These electrolytic ions will have sodium ions, we have hydrogen ions, and all of those ions, carbonate ions, sulfate ions. They help in pH buffering. They help to keep the pH of blood within the normal level that we said is around 7.4. Okay, so those ions help to buffer the pH and keep it steady. When one of these ions, for example, hydrogen ions, is missing from the blood, then it means that we would have alkalosis, or when we now have an excess of the hydrogen ion, we'll have acidosis. But the ions are there to maintain a steady pH of the blood. Good. So now let's take the first blood cell, which is erythrocytes, or the red blood cells. Now the red blood cells, they have a shape that is called a biconcave shape. So the red blood cells are like a disc. Okay, they look like a disc with a biconcave shape and a depressed center. So red blood cells are circular, biconcave, non-nucleated. So they do not have nucleus. They are void of nucleus. So remember that red blood cells are void of nucleus. They are non-nucleated. They have a biconcave disc shape with a diameter of 7 to 8 micrometers. The diameter of a red blood cell is 7 to 8 micrometers. Remember these things for multiple choice exams, because in objective exam questions, they're going to ask you, what is the shape of the red blood cell? Does it have a nucleus? What is the diameter of the red blood cell? They're going to ask you all of these. So diameter is 7 to 8 micrometers, and then the thickness is 2.5 micrometers. Now, the color of the red blood cell. Now, because of the pigment hemoglobin, which is in the blood, the red blood cell has a red color. So this red blood cell has a red color. This is the only blood cell, that has this hemoglobin pigment. And this hemoglobin, we will look at the function of hemoglobin later when we talk about the oxygen transporting function of red blood cells, because red blood cells transport oxygen around the body, and they are able to do that because of hemoglobin. But the hemoglobin is red in color, so it gives that red color to the red blood cells. Now, what is the amount of red blood cells that we have in a pinhead of blood, or in the adult male? So there are about 5.4 million red blood cells per microliter of blood. So when you take a microliter of blood, in an adult male, there are 5.4 million red blood cells. While in an adult female, there are 4.8 million red blood cells per microliter of blood. So a microliter of blood is just like a drop, just one pinch of blood. Yeah. Inside that blood drop, you have 5.4 million red blood cells. If that blood was taken from a male, an adult male, but if the blood was taken from a female, you have about 4.8 million red blood cells. So that means that there are more red blood cells in a man than in a woman. Okay.
Okay.
Now you can see my screen, right?
Yes, sir.
Good. Now, lifespan. So how long does a red blood cell live? From the day it is produced till the time it becomes old and it is destroyed is 120 days. So just like humans have a lifespan, red blood cells have lifespan too. And the lifespan of red blood cells is about 120 days or about 4 months. So it means that if red blood cells are produced today in the bone marrow, after 4 months or 120 days, they will be destroyed because they would have been old and done with their functions and they will be destroyed. Good. Now, the next thing we need to know is the production of red blood cells. Now, red blood cells are produced by a process that is called erythropoiesis. So erythro means red blood cells. Poiesis is just the process of production. So erythropoiesis is the production of red blood cells. And this erythropoiesis takes place in an adult in the red bone marrow. So you have your bone marrow, especially the long bones, the hip bone, that is the femur, and then the breastbone, the sternum, and the ribs. These are the major places where red bone marrows are, so red blood cells are produced in an adult, okay? And the process is called erythropoiesis. Now, in a child up to 5 years, the bone marrow of all bones produce red blood cells. So when a child, from let's say from 1 year to 5 years old, that child produces red blood cells in the bone marrow of all bones. And then in a fetus, a baby that is still in the womb that is not yet born, erythropoiesis takes place in the liver and in the spleen. Now pay attention to this. In a fetus, there are no strong bones yet. There are no bone marrows yet. So it is the liver and the spleen that produces the red blood cells. But in an adult, it is the red bone marrow of long bones. In a child, it is the bone marrow of all bones. But at the same time, in an adult, red blood cells are destroyed in the spleen. In a fetus, spleen produces. In an adult, spleen destroys. Okay, so pay attention to that. And remember that for erythropoiesis to take place, there is a hormone that is called erythropoietin. Okay, erythropoietin is the hormone released from the kidney that helps the process of erythropoiesis. So erythropoiesis takes place in the bone marrow, but the hormone that triggers it, that stimulates it, is called erythropoietin, and it is released from the kidney. Okay.
Yes, sir.
Now, increase in the number of red blood cells above the normal value that we talked about, 5.6 million in a male or 4.8 million in a female. When the red blood cells increases above that normal value, we have what is called polycythemia vera, okay? And then when there's a decrease in the number of red blood cells below the normal value, we have what is called erythropenia. Penia means reduction in number. Polycythemia means increase in number of red blood cells. Good. Now, functions of red blood cells. What do the red blood cells do? What is their function? So the major function of the red blood cell is to transport oxygen from the lungs to the tissues, and then to transport carbon dioxide from the tissues to the lungs. That is the major function of the red blood cells. Now, when you take in oxygen through your nostrils, and it goes to your lungs, that oxygen does not remain in the lungs. That oxygen combines with blood in the lungs, but not just the whole blood, it actually combines with the red blood cells. Why? Because the red blood cell, we said, contains hemoglobin. Now, hemoglobin has affinity, has attraction for oxygen. So what happens is that the hemoglobin in the blood, when it gets to the lungs through the pulmonary artery, yes, through the pulmonary artery, the oxygen combines with the hemoglobin to form what is called oxyhemoglobin. So that oxyhemoglobin is now transported through the pulmonary vein in the blood into the left atrium, from there to the left ventricle, and then it is circulated around the body. So the blood goes to the different tissues and releases the oxygen, break the oxygen from the oxyhemoglobin. So oxygen is delivered to the tissues, and then hemoglobin, goes back again to receive oxygen again to form oxyhemoglobin in the lungs, and then goes to different tissues to supply the oxygen. So the main function of red blood cells is the transport of oxygen from the lungs to the tissue. So it means that if somebody is having erythropenia, low amount of red blood cells, that person will definitely not have enough oxygen being transported to all tissues. And when oxygen doesn't get to all tissues, there can be no respiration, and there can be no cellular activity. There can be no oxidative phosphorylation, which you will do in biochemistry. Okay, almost nothing will function if oxygen does not get to your cells and your tissues, because those tissues depend on oxygen. Without oxygen, you cannot even get energy, because it is the combination of oxygen with glucose that leads to the production of energy, or ATP, adenosine triphosphate. So red blood cells are very important. Without red blood cells, we would all die. We cannot survive without red blood cells. Because once oxygen is deprived, what our tissues are deprived of oxygen, they begin to die. Okay, good. They will have what is called cell death. Okay, now the normal blood contains about 13 to 15 grams of hemoglobin. So this is HB, hemoglobin, per 100 mils of blood. So there are 13.15 grams of hemoglobin in 100 mils of blood. Now, one red blood cell contains about 250 million molecules of hemoglobin. One red blood cell contains about 250 million molecules, and each of these molecules will combine with oxygen to form oxyhemoglobin. And then when they get to the tissues, they deliver the oxygen to the tissue, and then they go back and take another oxygen from the lungs, deliver to the tissue like that, okay? Now, the structure of hemoglobin is that it has 4 molecules. Each molecule of hemoglobin carries 4 molecules of oxygen. Each molecule of hemoglobin carries 4 molecules of oxygen. So anytime hemoglobin binds with oxygen, there will be 4 molecules of oxygen bound to one hemoglobin level, one hemoglobin molecule. And that means if we have 250 molecules of hemoglobin multiplied by, each of them has 4 oxygen attached to it, 4 molecules of oxygen. So they are able to adequately supply all tissues of the body with oxygen. Now, let's go to the white blood cells. Remember, I said, even though I didn't show you, remember I said that after 120 days, the red blood cells are destroyed, okay? After 120 days, they are destroyed in the spleen. And once they are destroyed, the hemoglobin in that red blood cell is converted into 2 things. One is called ferritin, the other one is called bilirubin. Ferritin can be reused again because ferritin contains iron, okay? So hemoglobin is made up of hem is iron, okay? Hem is iron plus globin. So in hemoglobin, we have iron molecules and globin, which is a protein. But once after 120 days, when the spleen destroys the the red blood cells, the hemoglobin in that red blood cell now splits into two parts. The one part is called ferritin, which is the iron containing part and can be reused again, to produce more new hemoglobin. While the globin part, okay, is converted into what we call bilirubin. And later, when I teach you how bilirubin is metabolized, we will see that bilirubin can be converted into different things, can be conjugated. So we have conjugated bilirubin and unconjugated bilirubin, and each of them has their own role. Some are excreted, some are used to form bile, but we'll talk about that later in the future. Now, white blood cells. So the white blood cells are, oh, I just noticed that we exceeded our time, but I think let's go ahead. Or maybe we should do white blood cell another time. What do you think?
Maybe another time.
Yes, let's do white blood cells another time. So that today we know that we did red blood cells.
Yes,
and then in the next lecture, we would look at the white blood cells, and then we'll look at the platelets and the functions of each of them. Okay, good. So let me see. I need to, where are we? I can find a way to end my slide. Okay, good. So that will be all for today. We will meet next week.
Thank you, sir.
Thank you so much. Bye.
Yes, sir.